Are you telling me that I'm not good enough for you? No, it's not that. I'm just not sure if I'm ready to date again. I'm just coming off a bad mitosis. Now, if you've understood mitosis, I'm sure you understand how delicate this can be. Let's move on to our next session. This is a special type of cell division, and this is called meiosis. Now, if you were to define meiosis, you would say it is a special type of cell division which leads to the formation of 4 haploid daughter cells which are non-identical to the parent. So the hallmark is genetic variability. That's an important word associated with meiosis. And of course, it consists of meiosis I and meiosis II, and it is the first division, that is known as the reduction division, where the haploidy is induced, while the second division can just be a typical mitosis, except that there is no S phase, which means that after the first division there is no duplication of DNA. So a typical mitosis will just convert 1 haploid cell into 2 haploid cells, thus making a total of 4 haploid cells or gametes. Let's first study meiosis I. There'll obviously be a pre-meiotic S phase associated with DNA replication, as you can see here. And now a typical prophase I will have the characteristic coiling, supercoiling and condensation that is associated in mitosis, but it is long and it can further be divided into 5 stages. Let's talk about each and every stage. The first stage of prophase I is known as leptotene. Now, a homologous pair of chromosomes, like the one that came from the maternal side and the one that came from the paternal side, both of these try to search each other, and as soon as they find each other, they form a bond with each other. Now, that is the stage of zygotene, which is characterized by the formation of what are known as synapses. It can also be called synaptonemal junctions. You can see them happening here and here. So the light blue and the lighter blue are a pair of homologous chromosomes, and the dark pink and the light pink are a pair of homologous chromosomes, and you can see that now these chromosomes are connecting with each other to form synapses or synaptonemal junctions. As we head to the third part of prophase I, also known as the pachytene, this is where all such homologous pairs have now formed synapses with each other, and they continue condensing more and more to obtain a typical structure that is known as a bivalent. It's also called a tetrad, and this is typically what one bivalent looks like. Now, in the next stage, that is the diplotene stage, there's going to be something known as crossing over, and this literally means that one of the arms of a sister chromatid puts the arm around the other, and there is a certain exchange of genetic material. If you observe closely, you can see that happening. This is known as crossing over, and it is a characteristic feature of the diplotene stage. And finally, after the crossing over has happened, and the places where it happens are also known as chiasma or chiasmata, the sister chromatids once again separate from each other, but now they have changed slightly because each of these arms now have a genetic material of the adjacent arm, as you can see over here. So this is a phase known as dikinesis. It is also known as a latent or a resting stage. You'll see how in oogenesis the primary oocyte is arrested here until puberty is reached. Right, let's move on to the next steps, which are again same as mitosis, but here's a couple of differences. Firstly, in metaphase 1, yes, the homologous chromosomes as pairs do arrange themselves along the equator, but the splitting does not happen along the centromere. The splitting happens along the synaptonemal junctions. So this is the part where splitting happens, and thus each of these chromosomes now will go to their respective poles, which is a characteristic feature of the anaphase. Now remember, in anaphase, the chromosomes are randomly distributed to either side of the equator, and this is known as random segregation. So we already had a genetic variability introduced by crossing over that happened in the diploidine stage, and now once again either of the chromosomes can go towards either of the poles, introducing further variability, a process known as random segregation. Finally, now that the two cells are formed, remember that they are haploid in their chromosomal constituency and they do not resemble the mother cell. They are different than the mother cell. The most important thing now is that these cells, before entering meiosis II, do not have a S phase, which means they do not undergo DNA replication. So what follows is just a typical mitosis. So meiosis II is nothing but mitosis without the S or synthetic phase that is characterized before mitosis in otherwise normal cells. So you have these two entering prophase, again supercoiling, condensation, etc., etc., and this time in each of these the chromosomes are going to arrange themselves along the equator. So in metaphase 2, the splitting is going to happen along the centromere. Okay, and once the splitting happens, you have your typical anaphase 2 where you can see each of these going towards their respective poles, and now you have 4 haploid gametes, each of which are different than the mother cells. So to summarize meiosis, here are some of the features which can also be used as differentiating points from mitosis. Point number 1, 4 haploid gametes are formed in meiosis as opposed to 2 diploid cells in mitosis. Number 2, the cells that are formed are genetically different from the mother as opposed to mitosis. Number 3, in meiosis I, there is a reduction division that takes place, and that is where the haploidy is induced. In that, prophase 1 is long and can be divided into 5 phases: leptotene, zygotene, pachytene, diplotene, and diakinesis. In these phases, the coiling, supercoiling, and condensation continues, but also what happens is formation of synaptonemal junctions where homologous chromosomes come together, formation of chiasmata where certain segments of the chromosome cross over each other and exchange segments, thus inducing genetic variability. Then we go on to metaphase 1, which is different from the metaphase of mitosis in the sense that there is no centromere splitting. The chromosomes split at the synaptonemal junctions. Then we move on to anaphase 1, which is different because the split chromosomes might go to either pole randomly, thus introducing further genetic variation. And then meiosis 2 is exactly like mitosis, except there is no S phase that precedes mitosis. Thus, the haploid cells each will be converted into 2 haploid cells each. I hope that completes your understanding of meiosis, because now we are going to start getting a little more in depth of a process known as gametogenesis, for which the understanding of meiosis is absolutely essential. Thanks for listening, and I'll see you in the next one.